Dear students, now I am going to discuss about the development of dicot embryo of Capsula bursa pastoris of the family Brassicaceae. And this is the LS of the ovule of Capsula bursa pastoris just after fertilization. So here the egg has been converted into diploid zygote, and this one, that is the polar nuclei, has been converted into endosperm nucleus. In first step, this zygote cell will divide transversely to give rise to two cells structure, the lower larger basal cell, that is CB, and the smaller terminal apical or apical cell, that is CA. In the next step, CA and CB, both of these two cells will divide. That is, the smaller apical cell, it will divide longitudinally to give rise to A1 and A2, and the basal, that is the larger cell, it will divide transversely to give rise to C and D respectively. Thus, a 4-cell structure has been obtained. In the course of further development, A1 and A2, they will divide transversely to give rise to this 4-celled or quadrant structure, and C and D will divide and redivide to give rise to this 8-celled suspensor. The basal cell has been converted into vesicular cell and it will be grow further, and the apical cell is termed as hypophysis. If this quadrant and this suspensor, they will combine together here, it will be just like this. This is the lower part and this is the globular upper part. Now this part and this particular cell will divide and redivide to give rise to the dicot embryo. In the next course of development, these 4 cells, they will divide transversely to give rise to this 8-celled or octant structure. So the total structure has been converted into this. This is the suspensor and this is the 8-celled pro-embryo, globular pro-embryo. The octant cells, they will again divide periclinally to give rise to 16 cells structure, and these 16 cell structures, they will arrange like this: the outer layer and the inner cells. So the outer layer comprises of 8 cells and the inner one again comprises of 8 cells, and the hypophysis, it will divide to give rise to this particular structure. So the hypophysis will differentiates into the cortex and epidermis of radicle and root cap, and the basal 4 cells of the octant, by division, they will give rise to hypocotyl and the core of the radicle, and the terminal 4 cells of the octant, they will give rise to the plumule and the 2 Cotyledons. In the next step, the Cotyledons will grow further, and due to the growth of the Cotyledons from the ridges of the globular structure, the globular structure now will be converted into this heart-shaped structure. In the next step, the heart-shaped embryo will grow further. So the further growth will be resulted into this one: suspensor, root cap, dermatogen, peribulum and plerome, plumule, and the 2 Cotyledons. Now at the last stage of the development, the whole structure will be like this. This is the vesicle. These are the cells of the suspensor. So this is the root cap. This part is the radicle. This part is hypocotyl. This is the plumule, and these two, that is the C1 and C2, are the cotyledons. And due to further growth and development, it will push inside the endosperm cells, and it will be grow further and further, and it will be converted into horseshoe shaped structures. So this is about the development of dicot embryo in Capsula bursa pastoris. Thank you.